And then he did a postdoc at Kellogg Biological Station in Michigan with Jim Salao. And then his last year is being professor at Caltech. Now, one of the things that is really interesting about his work is that he uses a combination of theoretical and empirical approaches to looking at questions that are right on the interface of the and he's done this using a variety of different types of study systems, but the most interesting one is the feature plant is that allows you to actually answer a variety of questions on this question that is and today he's going to talk to us about evolution on the superset of the species.
Thank you guys for having me. You didn't boo or anything when she said USC. That was good. So I'm going to talk a bit today about this pitcher plant system that I worked on mostly for my dissertation, but I'm starting to work in a bit now, which doesn't really make any sense because it's a East Coast system and I'm on the West Coast now, but we can do a lot of stuff in the lab with the system, so that's really nice. So I'm a community ecologist, so I'm really interested in diversity. That's the one word I would use to sum up the things I'm interested in. So I'd be interested in looking at like a coral reef and thinking about why there's so many species there and how they all coexist together and what mechanisms allow them to coexist. So that's half of what I'm interested in. And then the other half is I'm interested in how these species evolve and how diversity affects that evolution. And so if you're, for instance, a coral growing on this reef, you not only have to deal with competition from other species on that reef, but you have to deal with, say, predation from fish and competition from algae. And there's a lot of different selective pressures for one species on that reef. And so I'm interested then in how those things combine, how evolution in response to those different selective pressures affects interactions between those species. So, so I'd say classically people that have done evolutionary experiments fall into one of two categories. In that one, there's these sort of in the lab bottle experiments where people manipulate some factor. So maybe you grow a plant in the greenhouse and you manipulate, say, herbivores on that plant, and you look at selection in response to those herbivores or evolution in response to those herbivores. And these are really nice because you can measure selection in response to some manipulated factor. And if you've manipulated herbivores, you can compare with and without herbivores, and you know that any effect you see is due to some extent to that herbivore. And so it's great for getting at a mechanism of what's driving evolution. And then the other class of studies fall into field observations where people go out to some field and maybe you just measure selection on this plant in the field. You measure fitness, you measure a bunch of traits on that plant, you do a selection analysis, and you look at whether there's selection on some of those traits. The problem is that when you do this, it's great because the good part of that is that it's selection in the wild. And so you understand selection in response to all the selective pressures that plant is experiencing. The bad part is you don't know which one was the most important. So you don't know whether really herbivores or competitors are doing selection on that trait. So the thing that I think ties these together a little bit is this evolution in a community context where we can start to look at mechanisms of what drives evolution in this community context when there's multiple direct effects on species and you start incorporating indirect effects. And we can start to get at some of the more complex mechanisms where these manipulative experiments look at simple mechanisms. We can start looking at more complex mechanisms that might explain why traits evolve in a certain way in the field. So I combine three different areas of research. One is looking at species diversity, and one is looking at evolution. So I would say that diversity has this effect on evolution, and field experiments have sort of gotten at that. These manipulative experiments have looked at how species interactions might affect evolution. And one of the ways that those that evolution in a community context might be a more complex process is because of these indirect effects. So if we think about two, say, competitors with each other, so species A and species B, they both have negative effects on each other. If we add a third species in there, say this is maybe a predator, so species C eats A and B, and it has negative direct effects on those. We can also start thinking about the indirect effects in this community. So if the presence of B causes C to eat more B, then there's less of B to compete with A. And so the presence of B is really having an indirect positive effect on A. And there's less predation on A if C is eating B. And so that's just one indirect effect in this community, but you can draw other indirect effects as well. And as you start adding more species into this community, you start getting more and more direct effects when there's more species, but you get exponentially more indirect effects because you can start getting these really complex chains of indirect effects where maybe the presence of E alters the interaction between B and D and that affects A in some way. And so indirect effects ecologically have been shown to be really important, and they're more important in diverse systems to get these more complex indirect effects. But we don't really have a good understanding of what their evolutionary impact is. So that's part of what I want to talk about today is the evolutionary effect of these indirect ecological effects. So that's part of what I'm going to talk about is how species diversity might affect species interactions through direct and indirect effects and how that might ultimately affect evolution. But then another aspect of what I'm interested in is how evolution might affect species interactions, particularly when that evolution happens in really short time. So rather than thinking about millions of years, thinking about more weeks to years. And so if we're thinking about something like elephants, then evolution's probably not going to affect interactions on any ecologically relevant timescale, at least not that we can observe. Because the generation times of elephants are just too long to really observe evolution in that system. It doesn't mean they're not important, it means that we can't really observe the evolutionary effect on ecology. But if we think about like another system, like a bacterial system, we know things like antibiotic resistance can evolve really quickly. It's makeup, it's not a real wound, it's just we know that things like antibiotic resistance can evolve really quickly on the course of, say, weeks or months because the generation times of those bacteria are so quick. And so you might think, okay, well, I don't work on bacterial communities, so why should I be interested in that? Except that bacteria and all these microbial systems are intimately involved with almost every other organism on Earth. So this elephant has bacteria living in its gut, and that affects how it processes plant food. And so if those bacteria evolve, it might change the way that that elephant interacts with the rest of its environment. So there's a few examples of these really rapid evolutionary changes in nature, and then people started to look for these more in the last decade or so. So here are bighorn sheep in Canada, and this is over about a 30-year time period. And so this is the breeding value for horn length. So this is just the amount of horn length that's attributed to inherited genes. And initially, the breeding value for horn length is really big. And over that 30-year time span, it's decreased. So in horn length in the population is decreased too because of human hunting pressure. And so humans are trophy hunting for the big rams with big horns, and so they're passing on their genes less frequently, and the smaller horn individuals are doing better in the population. And so you get this evolution of decreased horn size. So that's over 30 years, which is maybe several generations for those sheep, but it can happen even faster than that in just one generation. So this is an example with trout that are kept in captivity. The trait here on the x-axis is just the mean number of offspring per broodstock. The trait doesn't really matter so much is that if you look at 0 generations in captivity versus 1, these trait values change over that 1-year time span. So there's an evolutionary change in just 1 year in captivity. It's not always in the same direction. There's always a change over that 1 year. And so that suggests that these evolutionary changes happen really quickly in just 1 generation. Except these examples both deal with anthropogenic effects on communities. These are humans imposing really strong selection and driving evolution. And so does that really happen in nature? Can we, in a natural system that's not selection imposed by humans, does the same sort of dynamic happen? Do you get this really rapid evolution? So here's where I want to go, a short outline of the talk today. So one is looking at whether evolution alters species interactions. So that's looking at this arrow here. Does evolution affect the way species interact with each other? The second is, does diversity affect trait evolution? So does diversity somehow have an effect on interactions that then feeds back to evolution? And can we get this reverse pathway then? Does evolution affect species diversity? So if evolution affects species interactions and species interactions have some effect on diversity, then you might see a feedback there. So I've worked on these questions on a lot of different systems, which is both good and bad. I'm always jumping around from system to system. And so rather than talk about all those today, I'm going to focus just on these pitcher plant communities and some modeling work that we've done that addresses these questions too. So these are these pitcher plant communities. So they're a carnivorous plant, Sarracenia purpurea. It's, they make this little cup-shaped leaf that then fills up with rainwater, and then insects fall into that rainwater and they die, and then the plant consumes those insects and uses them for nutrients. So they tend to grow in really nutrient-poor soils where other plants can't grow very well, but carnivorous plants grow well there because they can get their nutrients from this outside source. And I'm not going to talk about the plant so much today as this community that exists inside of the leaf. So in that rainwater in the leaf, there's a whole community of microbial species in there. So they make this little food web, the bottom of the food web. Those ants are consumed by bacteria. Those bacteria are then consumed by a suite of several different protozoa. There's about 5 common species in there. There's also a rotifer that's endemic to pitcher plants. And then there's this specialist mosquito larvae that only exists in pitcher plants. So it only lays its eggs in pitcher plants, and it tends to be the only mosquito larvae you find inside of these plants. So here's just a little natural history of what happens over time in these leaves. So we went out and followed leaves from the time that they open until the leaf either died or didn't retain water anymore. So we're tracking these communities over time. So this is leaf age on the x-axis. Leaves can live up to a little over a year. I think the longest one we found was about 72 weeks. The average age is somewhere around 30 or 40 weeks. And so the first thing you notice is that all the dead ants are captured by young leaves. And then they decline over time because they're just being decomposed in the leaf. So those dead ants are captured in young leaves because the leaf produces a little bit of nectar along the lip of the leaf that attracts ants to it. And that's when it's doing most of the leaf capture. It only produces that nectar for the first like 2 or 3 weeks that the leaf is open. And so then it just stops producing that nectar and stops capturing prey. Occasionally it will act as a pitfall trap where like a spider or a grasshopper or something falls in there and you get this big nutrient pulse. But for the most part, all the nutrients are coming to the system really early on. You see bacteria follow that same pattern where you get lots of bacteria in young leaves and it just declines over time. You don't see a really strong effect with bacteriivores. So these are the protozoa and rotifers. Maybe there's a little intermediate peak in abundance there, but it's not a really strong pattern. And then mosquitoes go back to this other pattern where there's lots of mosquitoes in young leaves and not very many mosquitoes in old leaves. And those different colors are just different instar levels of mosquitoes, but they all follow the same pattern. And so what's happening is all those mosquito mothers are laying eggs in really young leaves. You get lots of predation there. Mosquito larvae grow up and hatch out and fly away. So what that means is if you're a bacteriivore in this community, that at really young ages of leaf, there's lots of predation, but not very much competition because there's lots of resources and species don't have to compete with each other. In these really old leaves, there's not a lot of predators around, but there's not very many resources, and so we expect that competition might be really high in those really old leaves. So for the first part I want to talk about, I'm just going to focus on 2 species in this leaf. One is Colpoda. It's a ciliate. It's kind of intermediate in size to all the other bacteriivores in that community, and it's consumed by this mosquito larvae. So this is Wyomia smithii. And so we just do some lab experiments looking at how Colpoda responds to those mosquito larvae and whether that changes over time. So we start with just a stock culture of the prey, so that's the Colpoda. And we introduce as much genetic variation as we can into that culture. We don't know how much genetic variation is really there, but we try to maximize it. We take some of that stock culture, put it into either a no predator control or put it into a predator treatment where there's 2 mosquito larvae in this treatment. And then this is done in 5 different time blocks with different levels of replication in each one. But in each one, we let these things evolve for about 6 days. So that's around 30 generations. So a generation time in these is somewhere between 4 and 8 hours, so roughly 6 hours. So we're getting about 30 generations of evolution in there. And then we throw them in a common garden with the idea that if you just measure traits in this treatment and this treatment, it could be due to evolutionary change, but it also could be due to some phenotypically plastic response too. And so if we take organisms out of these treatments, put them in some common garden where they're experiencing the same environment, then we measure traits in that environment. And so then we know that any differences between treatments are due to evolutionary change and not a plastic response. So measuring traits is not super easy in these things because they're microbes and you can't see them very well. So these are the 6 traits that we've tried to measure in these. And I've grouped them into resistance strategies and tolerance strategies. So the resistance strategies are things that allow them to resist predation or avoid getting eaten. The tolerance traits are things that allow them to just keep up with the rate of predation. So this is a little refuge we provide them at the bottom of the tube. So we put these little beads in there. They mimic all the frass and insect exoskeletons that you normally find at the bottom of a leaf. And if we don't put that in there, we find that the predators consume all the prey in about 24 hours. But if you give them this little bit of refuge at the bottom, they'll persist for weeks at a time. And so the predators are still eating down this refuge, but they're not foraging as efficiently. So it might be that these cells increase their use of this refuge. They could change their swimming speed, they could change their cell size, they could change their population size, which we assume to be indicative of like their resource use efficiency, so this is presumed to be a proxy for their carrying capacity. We can measure the growth rate of the population, or we can measure, they make these little cysts that allows them to survive harsh conditions. So maybe they make more cysts when there's predators around. So I'll just show you a couple of these traits. So this is population growth rates. This is how fast the population is growing. We originally started thinking about this as a trait that it would be like little r, like the maximum amount that a population can grow. Now we're a little uncomfortable thinking about that as a trait and think about it more as fitness. And so these are cells that were taken from each of these 2 selection environments. So cells that were taken from an environment that had a predator grow faster than cells that were taken from an environment with no predator. So they have higher fitness. And this happens whether you're growing them with a predator or without a predator in the common garden environment in which we measure them. So the ones that evolved with a predator always end up with higher fitness than the ones that did not evolve with a predator. So presumably there's a trade-off with something else in that environment, maybe competitive ability or something, but we haven't quite figured that out yet. If we also look at cell size, so this is a better trait to look at. Cells that evolved with a predator tend to have smaller cell sizes than those that evolved without a predator. And we think that's because, at least across species, predators prefer to eat bigger cells and they avoid smaller cells. And so that might also happen within a species as well. That maybe within a species, the bigger genotypes are getting eaten, the smaller genotypes are surviving better. So we find no effect of the predator on evolution of these other traits. So it seems like they might be evolving increased growth rate, which would be a tolerant strategy, and that they can just keep up with predation. They might be evolving a smaller cell size that allows them to resist predation. They just get eaten less often. But then what we're really interested in is whether that evolution of traits affects the interactions between the predator and the prey. So we measured interaction strength as just the difference in abundance between the 2 treatments, standardized by the monoculture. So this is the predator interaction strength. So a value of 0 would suggest the predator has 0 effect on the prey population. And then negative numbers mean the predator has a negative effect on the prey population, like you'd expect with a predator and prey. And if we look at day 1 of the experiment, predators reduce the prey population by about half. So having a pretty big effect on the prey population. But then that changes over time, so that if you look by like day 11 or 12, the predator is having no effect on the prey population anymore. And it's not that the predators are eating less, because we replace the predators every other day. So they're still hungry and they're still eating cells in there. They're just not having an effect on the population. And so what we think is happening is that the predators might still be eating the same amount, but the prey are just keeping up with that predation. So if they're dividing at a faster rate, they're just dividing quickly and keeping up with that rate of predation. And so you get sort of a coexistence strategy here where the predators are happy, they're still getting all the food they want. The prey are not being affected very much because they're just keeping up with all that predation. So predators cause the evolution of decreased cell size in the prey, and the evolution of that cell size or growth rate reduces the ecological effect of the predators. And I think what's interesting about this is that the interaction strength between the predator and prey would be misinterpreted if you just looked at the end of the experiment. So if you just looked at the end of the experiment, you would say, this isn't a predator-prey relationship at all. Like, these predators are not eating this prey. But it discounts the fact that the predators drove this evolution of these traits that allowed them to get to this zero interaction strength. So I think about this as the ghost of predation past, that the predation was really important in driving the evolution of these traits, but you don't see that, you don't see a signal of that interaction anymore in a contemporary community. Okay, so does evolution alter species interactions? Yeah, at least for predator and prey, there's a big effect of evolution on those interactions. And now we want to know whether diversity affects that trait evolution or not. So that was a pretty simple community. There's just one predator, one prey species. And obviously these things are existing in an environment where there's lots of competitors and other things around. So we also did the exact same experiment we did with this predator, but with another competitor. So this is Colpidium, which is a similarly sized ciliate. So we do the exact same treatments and allow evolution in response to the competitor, with the thought that we'd get a trade-off here, that traits that evolved in one way in response to a predator would evolve the other way in response to the competitor, and there'd be some trade-off between predator tolerance and competitive ability. And we didn't find that at all. We found that these same two traits evolve in the same direction. So you get increased population growth rates, decreased cell sizes, you get a little bit of evolution of refuge use, and a decrease in cyst production when cells evolved with competitors in the past. So if there is any evidence of a trade-off there, it's not in response to this particular competitor. It doesn't mean there's not a trade-off in response to some other competitor. And so now we wanted to look at, so that was just what happens when there's a predator there, what happens when there's a competitor there, but what happens when there's both there at the same time. And so now we still use a simplified community, but it's a little more complex. And I think the key is adding this third species, because now you not only get direct effects from predators and competitors, but you also get indirect effects. So now these predators are eating these competitors also, and so they might have an indirect effect on our focal species here. So we did a similar experiment to what we did before, where we have cells growing in monoculture. We have cells growing with 2 mosquito larvae, predators. We have cells growing with competitors, the same competitors before. And then we have cells growing with predators and competitors. And the idea here is that in this last treatment is where you get direct and indirect effects. So that's problematic in terms of trying to partition those direct and indirect effects. But because we know the direct effects from each of these treatments, we can subtract out the direct effects from here, and we're left with just the indirect effects from that total effect. So we let these things evolve. I think for this case, it's for 20 days, so maybe 100 or so generations. We again take cells from each of those treatments, grow them in a common garden, so we get rid of any plasticity effects, and then measure traits again in that community. And so this is not real data. This is just made up data. So this is if we have some trait value on the y-axis, trying to partition out those indirect effects can be a little bit tricky. So I just wanted to walk you through how we do it. So we have cells that evolve in a monoculture, say, have this value. We can measure the direct effect of the predator as just the difference in effect size between the monoculture and the predator treatment. And we can measure the competitor effect as the difference between the monoculture and the competitor treatment. And then if those effects are additive, if those species don't induce any indirect effects, if we're just adding their effects together, we should see this. We should be able to add the red arrow to the blue arrow. And if the effects of predators and competitors are purely additive, if they're not interacting with each other, we'd expect a very large trait value when both species are present. And any deviation we see from that has to be due to some indirect effect. So we can measure that, the non-additivity, as the difference between that predicted additive effect and the actual observed value we see in that community. So here's actual data. So this is cell area again. So we see the same thing we saw before, that cells that evolved with predators are smaller, cells that evolved with competitors are smaller. And so what you might expect is that if you add those effect sizes together, that you get the evolution of very small cells when both predators and competitors are present. And instead, it's not what we see. So we see cells that evolved with competitors and predators have the exact same cell size as those that evolved in monoculture. And it's because of this really strong indirect effect. So something's going on that those predators and competitors are interacting with each other in some way that causes an indirect effect and changes how that population evolves. So here's what I think might be happening. So we do know that this is our focal species Colpoda. They partition the water column a little bit, and that the bigger cells tend to stay up in the water column and the smaller cells tend to stay down in that refuge. And so if you add a predator into that community, they can forage really efficiently up in the water column. They don't forage very efficiently down in the refuge. And so the cells that are experiencing a greater deal of the predation are the big cells that are up in the water column. And so you get, what you're left with is a population of small cells and not very many big cells. If you add a competitor, the same thing happens. So the competitors, for whatever reason, stay up in the water column. So you tend to find most of the bacteria down at the bottom. They all sink to the bottom. So there's a lot more food potentially down at the bottom of the tube, but it could be that different bacterial species partition the water column differently too. And so potentially these prefer some bacteria that's up in the water column. But for whatever reason, the competitors tend to stay up in the water column and not in the refuge. And so then the individuals that are experiencing the competition are those that are up in the water column. And so again, what you're left with is a population of mostly small cells that live down near the bottom. If you add both those species in together, they effectively cancel each other out. So the predator prefers to eat this competitor. And so the predator is happily eating the competitor and not eating very much of the focal species. At the same time, the competitor population is now reduced because they've all been eaten, and so they're not having a very big competitive effect on the focal species. And so effectively, these two species just wipe each other out, and you end up with no net selection in response to either one. And so they're both having this really positive indirect effect on Colpoda. So in this case, diversity does seem to affect trait evolution. If we go from a 2-species system to a 3-species system, those indirect effects you see in that more complex system change the way that population evolves. We don't know what happens if you go to a 4-species system. You might get even more complicated indirect effects that could potentially change the story back in the other direction. But in terms of just this simple 3-species system, the indirect effects we see there are just as important as the direct effects. So the indirect effects completely offset the direct effects. So they seem to impose strong selection in the opposite direction. They might play a role in maintaining genetic variation in that population. So if you go back here, you can see here you're left with very little genetic variation, here very little genetic variation, but in this population, potentially you're left with a lot more genetic variation. And so it might allow those populations to still respond to selection in the future if there's still a lot of different variants in genotypes there. And then the last thing I wanted to point out is just that evolution in the wild occurs in this community context. So very few species evolve in isolation, just interacting with one other species. Most of them interact with dozens of other species over their lifetime. And so evolution response to these indirect effects could be really common in natural environments. So one of my grad students now, she's doing a similar experiment, but rather than looking at biotic interactions, she's looking at abiotic interactions associated with global change. So she's looking at increases in temperature, increases in nutrient inputs, and thinking about the same sorts of things is whether you could have non-additive effects of these global change factors that might affect how species evolve. So she's starting again with a genetic stock of protozoa, growing these in these different combinations of environments, and then looking to see whether the effects of those things are additive or non-additive. Whether they're they have non-additive or additive effects on selection. Okay, so that was the second part. So does diversity affect trait evolution? Yeah, it does, through indirect effects. And then the last bit I want to talk about is how evolution affects species diversity. So do you get this feedback where evolution changes species interactions and then ultimately feeds back on diversity? And I'm going to talk about this more in a modeling framework rather than with that same pitcher plant system. So to switch gears a little bit, so niche theory, classic niche theory that underlies most of ecology, suggests that there's some resource gradient and that each species uses a different portion of that resource gradient. So if this is, say, seed size on here, you have the blue species using small seeds and the green species using large seeds, and that species coexist because they use different niches. And then maybe you've heard about this around the hallways, that neutral theory suggests that species might coexist because they're not any different from each other. And so as long as you could have species using exactly the same resources, and if they have the same fitness using those resources, that they might coexist. So niche theory always predicts that these species should diverge away from each other to partition the niche even further. And neutral theory suggests that they might not have to partition this niche at all. And I'm not going to go into the whole debate between niche and neutral theory, but we wanted to ask whether this condition could even exist and whether you might go from this condition to this condition. So are there situations in which species might converge in their resource use? So increase their niche overlap rather than decrease their niche overlap. So originally we were thinking about this, it didn't make a lot of sense to me. So if you have some species where there's significant niche overlap and some difference in their competitive ability, there should be two potential outcomes. That niche theory suggests that you should either get extinction or you could have some level of character displacement where species partition the niche, and that leads to coexistence. And then back in the 80s, Lonnie Arsen suggested that there's a third potential pathway there, in that you could get species to converge with each other, and that could be another path to coexistence if species become equivalent species. And that didn't make a lot of sense to me until Mark McPeak explained it to me this way. He said, imagine a boxing match where you have two really unmatched competitors, right? So if you have Muhammad Ali fighting Pee-wee here, the outcome is obvious, right? That Pee-wee's going extinct in that system. And so if Pee-wee engages in that competition, he's going to go extinct. And so the best strategy for Pee-wee might be to just avoid that competition altogether. So undergo some character displacement where he avoids any confrontation with Muhammad Ali. The other situation would be if you have two really well-matched boxers. Then you could get coexistence for a long time period because those boxers are so well-matched. And so eventually maybe someone's going to win in this scenario, but it's going to take a long time for that, for any extinction to occur. And if they're perfectly matched with one another, then you might not, the match could go on for an infinite amount of time. So we created this model just to sort of model that dynamic and see when and how each of those pathways might occur. So the math is pretty simple in here, so I'll just walk you through it a little bit. So we start with this U matrix that describes the use of, in this case, 3 different resources by 4 different species. So species 1 is using 70% of resource B, 20% of resource A, and 10% of resource C. So we can describe the resource use of each species. And then based on their overlap in resource use, we can make a per capita interaction matrix. So these numbers just describe how much niche overlap there is between species. So here, for example, species 1 and 2 are both using mostly resource B, and so they have pretty high interaction coefficients in that matrix. Versus species 2 and 4, 2 is using mostly resource B, 4 is using mostly resource C, and they have really low interaction coefficients in that matrix. And then we take the elements of that matrix and they get plugged just into a basic Lotka-Volterra growth equation where those are the alphas. So they describe how much the per capita effect of one individual of one species on another species. So that's the ecology of the model. So we can go from resource use to population growth. So that's how the ecology of the model works. The evolution comes in that we allow species to change their resource use. So each iteration of the simulation, each species tries each potential resource combination that it could, and the one that is the best resource combination is the one that's chosen in the next generation or selected for. And so that it's a system where there is assumed to be lots of mutations and then quick fixation of those mutations. So for instance, in this case, species 1, which had a lot of resource overlap with species 2, it starts using less of resource B and starts using more of resource A. And so that's the best strategy for species 1 to maximize its fitness in the next generation. And then we can change the rate of evolution here. So this would be a really high rate of evolution where a species in each generation can change its resource use by 20%. So we can change that rate of evolution and crank it up or slow it down. Okay, so here's what happens in the model when we just have 2 species. So this is also only with 2 resources. So that's so we can nicely draw on the y-axis the proportion use of resource A. So a value of 1 means that the species is entirely using resource A. It's a specialist on resource A. Or a value of 0 means it's using none of resource A, which means it's entirely using resource B. And then we can track their resource use over generations. And what we find is they partition the niche, that the green species starts using resource A, the purple species starts using resource B, and that niche is completely partitioned. So exactly what we would expect from niche theory. And then if we follow their population dynamics over time, so this is now abundance on the y-axis, they both do this logarithmic growth. They go up to some carrying capacity, and they're stable at that carrying capacity. So the interesting dynamics happen when we add a 3rd species into this system. So now we've added this red species, and the red species evolves a way to use resource B. The green species still evolves to use resource A, but now the purple species is kind of stuck in the middle. And so it, in this case, converges with the green species and starts using resource A. And so we do get some convergence there. And it's not that, so if we look at the population dynamics, the red species is doing really well because it's sitting at the carrying capacity for resource B, but the purple and the green species have now partitioned that carrying capacity for resource A. And so purple's not doing very well by converging with green, but it's still better than going extinct. So it still makes it there, and once it's made it to this point where it's an equivalent species to the green one, it can't outcompete the green one any more than it can outcompete itself, and vice versa. So they're effectively equivalent species, and they're coexisting there. So we can also change where we start species on this y-axis. So here species start out very similar to one another, but we can change where we put that purple species and look at the fate of that purple species based on where we start it on this y-axis. So that's what's here. So on this y-axis is the difference in the initial resource use. So this would be very similar species. These would be very different species. And this is just the fate of that purple species there. So, and then on the x-axis here is the rate of evolution. So this is slow evolution, fast evolution, relatively fast. What we find is that when the rate of evolution is really low and species are really similar to one another, you get extinctions. But if species are different enough, even though the rate of evolution is low here, species coexist because they're already pretty different from one another. But there's a point at which if you crank up the rate of evolution, extinctions just go away because species start coexisting with each other because they become equivalent species. So they evolve fast enough to become equivalent to one of the other species. And the difference between this area and this area is just which species the purple one's converging with. Either it's converging with the red one or with the green one. But either way, it's effectively the same result. You always get some combination of convergence and divergence. But in both of these cases, you get 2 species converging with each other. And so the point I want to make here is that you can get convergence, but it's always as a function of divergence. Species are always trying to be more different from one another. It's just if you're trying to be more different from that red species, you end up being more similar to the green species. and you've only got 2 directions to go, so you choose the lesser of 2 evils, which might be convergence with another species. And so in this case, I think what happens is that if evolution is really slow and species are really similar to one another, that's when you get competitive exclusion. But potentially, if evolution is fast enough, you can get coexistence, and the pathway you take to coexistence depends on whether those species start really dissimilar from one another. If they're already different from one another, it makes sense to continue diverging. If they're already pretty similar to one another, it's a short evolutionary pathway to become equivalent species to one another. So often that can be a path to coexistence. So this model suggests that species can evolve to be equivalent, but it only happens when the number of species exceeds the number of resources. So there we had 3 species using 2 resources. If we added in a 3rd resource, that purple species would go to use the 3rd resource. So it only happens when you have more species than resources. And it requires evolution in this community context where there's indirect effects going on there. So it's indirect effects that are driving one species to converge with another one in the process of trying to diverge from the first one. So that's one point. The second point is that rapid evolution increases coexistence, but it only happens when adaptation happens faster than competitive exclusion. So you've got to crank up that evolutionary rate fast enough so that the evolution's happening faster than the ecology of the community. But remember that as those species are becoming more and more similar, it's taking them, competitive exclusion takes longer and longer. Those boxers are becoming better and better matched. And so competitive exclusion takes longer to play out. So as they become more similar, the ecology is slowing down. And so the evolution, it might be more time for evolution to occur there. The other just kind of interesting thing we pull out of this model is that if we just throw 10 species randomly onto this y-axis, we find that some species go extinct. This one goes extinct, this one goes extinct. But you get about 4 species converging to use one resource and about 4 species converging to use the other resource. And so this might be one way where you can get the evolution of functional groups, is that here you have functional group A, here you have functional group B. And so it might be that that niche processes are driving the differences between functional groups, but within a functional group, you have neutral processes that are governing abundances and diversity within a functional group. So there's an undergrad at CSUN who's kind of taken up this model, and you'll notice that even in this situation where you throw 10 species, everything evolves to be a specialist. You never get the evolution of generalists in that model. And so Sam's taken this on and trying to see if he can tweak this model a little bit to a scenario where we could get the evolution of generalists. And I think one of the problems with this model is that we don't model the resources explicitly. So there's a constant supply of resources in that community, so it's like a chemostat sort of system. And I think modeling those resources explicitly might allow some species to be a generalist here in the middle when the resources are changing as resource use changes. So what then we want to know is if a generalist strategy can evolve, if you can have a species that stays here in the middle, can you also get convergence on that generalist strategy or not? Is there just then 3 strategies instead of 2? So we also wanted to test the ideas from that model in a real community too. So we went out to pitcher plant communities with the intention of testing this model. And so our thought was if we just look at 4 species, so we take the 4 most common species in a community and we measure their interaction strength with one another. So we take them out of the field, we bring them in the lab, do just competition experiments between them and measure their interaction strength, that we might expect, in the case of the green boxes, we expect these values go from -0.5 to -0.2 to 0. And that's if species are diverging, they have less and less resource overlap with one another. And so they might partition that niche so that they have no interaction with one another. And so in the case of the green boxes, that's where we expect divergence. We'd expect interaction strengths to decrease over time in the leaf. In the case of the red box, we have interaction strengths increasing over time. So that would imply that species are converging with each other. They have more and more resource overlap, and they're sharing a niche more, so they interact with each other more strongly. So we just wanted to go out into a community and ask this, do interaction strengths decrease or increase over time? and that might suggest whether species tend to diverge or converge. And so we did that. So we started with, this is a chrysomonad, this is Bodo, this is one species of Colpoda, this is another species of Colpoda. And we take them from 2-week-old leaves, from 7-week-old leaves, and from 12-week-old leaves, and we just measure their interaction strengths in the lab when they're taken from all these different evolutionary histories. And what we find is by the end, we find the ones that are really good competitors tend to decrease their effect on other species, and the ones that were initially really bad competitors tend to increase their effect on other species. So the good competitors are becoming worse, and the bad competitors are becoming better. And so they're all converging on this intermediate competitive ability. And so we don't know how to explain that. We expected that all the species should do one or the others, not that some species would do one thing and some would do the other thing. And so neither niche nor neutral processes really explain this pattern. So just maybe we need to think about what does niche really mean, and how do species really partition a niche, and what does that mean? So now we're trying to follow up, trying to explain this pattern a little bit better. So the next couple things we're trying to do is one, we're trying to look at whether evolution over this rapid timescale has an effect on the stability of communities. So not just interactions between species, but the stability of the community as a whole. So I'm working with Sebastian Schreiber, and we're using this pitcher plant system along with some simulation models to look at, as we allow species to evolve, does that increase or decrease the stability of the community, and does it depend on the types of interactions in that community? So we're taking communities that were made up where you have apparent competition as the dominant interaction, or exploitative competition, or omnivory, and asking, both in simulation models and in experiments, if you allow species to evolve in that community, does it increase or decrease stability? So that's one thing. And then the second thing we're interested in is generation times. So I mentioned at the beginning that you don't expect to see these evolutionary effects with elephants, but you might with bacteria. And so differences in generation times between interacting species are really common. That you have, say, annual plants being eaten by mammalian herbivores, or you could have really long-lived plants that are being eaten by insect herbivores with really short time spans. And so it might be really common that generation times differ between organisms. So I'm working with Mary-Alice Koffroth to try to do some of this work now, along with Shannon, one of my graduate students, where we're thinking about corals and symbiodinium that grows inside of corals, with the thought that corals are probably too long-lived and generations are too long for them to really rapidly adapt to climate change. But for these algae, a generation time is about a day or so. And so they might be able to adapt to that climate change, and by their adaptation, confer some adaptation to their host as well. So we have a little bit of preliminary evidence from the lab. This isn't a significant pattern, but the trends in the direction that one might expect is that if you take cells that were grown in a control environment or cells that were grown under heat stress, and then you expose them to heat stress, the ones that had previously been exposed to heat stress tend to have higher fitness than the ones that were not exposed to heat stress before. So potentially there's promising things happening there. So what I want to leave you with is just that with this puzzle analogy, that to me, putting together a community is like putting together a puzzle, that you have all these pieces that need to go together, and you're trying to figure out how these pieces all fit together into some big picture. And to see that big picture from a community ecology standpoint, you have to think about how those puzzle pieces might be changing. If the shape of those pieces are changing, that's going to affect the way the big picture fits together. And similarly, from an evolutionary biology perspective, if you want to understand what's going on evolutionarily, the more pieces of the puzzle you have, the better you're going to be able to put together that picture. So this is just people that are in my lab now, and a little bit of time for questions, if you have any. Yeah?
I didn't quite understand your description of the results that you got in your experiment where you attempted to test your current displacement model. So you basically said that the result didn't fit the predictions in the model, but I didn't really understand what the result was.
Yeah, so what we thought is that you have species with different competitive abilities initially, right? And it's based on size for the most part, that the big species are really good competitors, the small species are really bad competitors. And so we thought that the big and small species might just continue to partition the niche somehow so that they would decrease their interaction strength with one another. And so you'd expect those interaction strengths to just go down over time. But instead what's happening is that the big species are having less and less of effect on the other species, but the really small species are having a bigger and bigger effect on the other species. So they're not changing their morphology. It's not that cell size is changing, but for some reason their effect on the other species is changing, and their response to the other species is changing. So what you get at the end is a whole bunch of species that kind of have an intermediate competitive ability. They're sort of affected by other species, and they sort of have an effect on others, but not big ones.
How did you determine that that was what was happening?
That's, so we just measured their competitive ability each one of those times. So we measure competitive ability in really young leaves where, so that's sort of our time 0. And then at 12 weeks, they've evolved for 12 weeks out in nature. And then we take them out of those systems and then measure their competitive ability again. And they've changed, but not in the way that we predicted, but not exactly in the way we predicted. Instead, you kind of sort of get like this sort of communist system where everyone's equal at the end. Yeah. Priyanka.
So I have two questions about the model itself. So you have perfectly substitutable
Yes. Yeah.
and if you didn't,
Yeah.
you get a different outcome.
I think so. That's part of what Sam's thinking about tweaking with this model a little bit too, is just what if, so there's some other models out there with non-substitutable resources. So Jeremy Fox has one of those, and he gets convergence in his model when the resources are non-substitutable. But I think it'd be interesting to look at non-perfectly substitutable. So maybe one unit of resource A gives you more calories than a unit of resource B or something like that. So that might be one way that you could get an evolution of generalists too. But I think it would still require modeling those resources explicitly to see what's going on with them.
And then my second question is, so, this idea of limiting similarity, there's
Yeah.
a minimum difference between species, and we don't see that in this model. Is it because you lack some sort of constraint? Say it's, I don't know, it's beak size or something, you really can't evolve it down to the level of the next smaller species. Is there something, because I couldn't quite tell since I haven't seen the details of it,
Yeah.
but is there something that, because usually you have some constraint driven by a trade-off or something that
Yeah.
prevent. It would be interesting to ask why you don't get that, and is that just because you have substitutable?
Exactly.
Then that would transfer to the print system where maybe they're not substitutable.
Exactly.
Right? So that's an interesting thing. I can't explain the results through that.
Yeah.
On the top of my head, but.
Yeah, I mean, so one way that you might get, not necessarily limiting similarity, but one way you might get some limits to evolution is we assume that all those species have the ability to evolve to be whatever, they can evolve anywhere in the landscape. But that might not be possible. If you're a large bird, you can't evolve a small beak because it's got to fit your body size.
I wonder if you could put in some, when each species or individual selects its best strategy for the next generation, if you put in some constraints, you can only do so much.
Yep.
Or something.
Yeah. I think that might be.
I'm just throwing this out.
No, that might be a way to get generalists in that model too, is that if you added a predator, and so maybe you can evolve to be a better specialist on some resource, but that comes at a cost of being eaten more often or something. And so then maybe there won't, right now there's 2 adaptive peaks, and you're always being selected towards the top of one of those adaptive peaks. But there might be, like you say, constraints or trade-offs that either prevent you from climbing a peak or make more adaptive peaks. So maybe there's another adaptive peak with intermediate competitive ability and intermediate predator tolerance or something too. So that's interesting. I haven't thought about adding a non-competitor in there, but that might be an interesting thing to do. Yeah.
I'd like to talk about actual picture plants.
Yeah, okay.
So your experiments focused on what happened during the course of species interaction within a single leaf.
Yep.
In nature, of course, you've got pitcher plants all over the place.
Yep.
Some of them are even at a distance from others.
Yep.
And somehow they get colonized by those species.
Yep.
And I'm wondering if And you didn't talk at all about the colonization process. And it may be that features associated with colonization are maintaining features in phenotypic traits within the populations that you don't know about yet.
Yeah.
And can you comment on that?
Yeah, so that's exactly where we're thinking about going with some of this stuff too is, well, one, I'll say, we don't know how colonization occurs. We've gone out and tried to measure it, and either we're really terrible at measuring stuff, or they're dispersing in a way we haven't thought of. So we've tried so one way is that they're just blown in by the wind. They have spores that blow in the wind or something. Another way is that maybe mosquitoes pick stuff up when they're flying from leaf to leaf. Another way is that maybe ants crawl in from the soil and they've got stuff on them when they fall in. And so we've tried putting tanglefoot around the bottom of leaves to prevent ants from getting in. We've tried putting bridal veil over the top of leaves to prevent mosquitoes from getting in. And we've tried doing neither and just allowing wind pollination or wind dispersal in there. And in every case, we've tried this by sucking everything out of a leaf, pouring a bunch of hydrogen peroxide in there to kill everything off, rinsing it out. And in every case, we get colonization really quickly. And so either colonization rates are really high or we're bad at sterilizing leaves, and I'm guessing it's the latter. So that's one thing, is we've tried to figure out how colonization is occurring, but we don't know. But the second thing is we don't know the rates of colonization, which would be really interesting. So maybe a leaf opens and all the things that are ever going to be in that leaf colonize it immediately, and then you just have ecology and evolution happening. Or maybe there's constant rain of gene flow into that community, which might affect particularly the evolutionary pathways too. If you constantly have this rain of gene flow coming in, but then it's going to matter where the gene flow is coming from. So if it's mosquitoes doing the dispersal, they're going from young leaves to young leaves. And so they're only going to move around things that might already be pre-adapted to the environment to which they're going. If it's ants going around, they could be bringing in particles that are maybe adapted, maybe not adapted to that community. So I think where the gene flow comes from is really important. So that's what we've thought about doing, both from a modeling and an experimental standpoint, is think about these things like a meta-community. And they're really set up well for that in nature. They are these nice discrete patches. And what happens when you allow gene flow, it doesn't matter where the gene flow is coming from.
Have you tried your artificial containers, putting those in the field?
We have, and they just get swamped with bacteria right away. So if we put out and this is in Florida, so it's hot and gross and there's bacteria on everything. So if you put out a tube of water with a couple ants in it and you come back the next day, it's just choked out with bacteria.
So the bacteria get there a lot faster than your protozoan.
Exactly, yeah. And so I think if you started with some protozoan there already, they'd probably keep the bacteria population down, at least to some extent. But they're probably not just blowing in on the wind if you just put a tube of water out and they're not making it there. So it suggests that maybe there's a biotic agent that's like, I'm not going in that plastic tube. So, hey.
So neutral theory, for example, predicts species event distribution, and two allows for things like connectivity. Your results here are very convincing, but have you thought about the extent to which they would or could change predictions like that?
About whether the stochasticity or something would change that? Yeah, so what happens in our model is you make it to be equivalent species and then you coexist there forever. But there's no stochasticity in that model at all. So what really should happen is that they should make it there, but then there should be some drift where one of them eventually goes extinct. And so they shouldn't coexist neatly at that carrying capacity, but one of them should slowly drift to extinction. And that would be interesting too, because that's not just going to affect the ecology, it's going to affect the evolution too. So if you're sort of drifting away from, say, a particular resource use, then it takes some selection to drive you back in that direction. And particularly interested in if there's strong selection in one direction, you might decrease the genetic variation during that selection process, and so you might not always have the ability to evolve back in the other way. So you might be stuck, and there might be some constraints there eventually if you start changing the genetic variation. Cool. Thank you, guys.
That was great.
Thanks.
A couple things. First,